Yeah, before we continue with the Aceolomate bilatarians, as you have seen the explanation in the previous class, just we are trying to see how it is given in the text here. So before we go with the Aceolomate bilatarians, we have a small question here in the box item. The pseudocoelom is considered as a false body cavity. Substantiate the statement. Yes, we can substantiate the statement. In what way? Embryonic blastocoel, which is present in the gastrula stage, embryonic blastocoel, we said that remains in the adult also. That embryonic blastocoel or primary body cavity remain in the adult also. To say true body cavity, which body cavity required? Secondary body cavity, which should be formed from the mesoderm. Secondary body cavity, which should be formed from the mesoderm, is required. But in case of pseudocoelomates, we have seen earlier, the mesoderm went only towards the ectoderm, and this cavity remain in the adult. That's why being they have the primary body cavity, the false body cavity, we consider it as a false body cavity. Or in other words, the cavity which is not lined by mesoderm, we can say false body cavity. So whom we can call true body cavity? You see here, this white space is secondary body cavity. What is the evidence that is secondary body cavity? That we have an evidence. You see, both sides that is lined by mesoderm. That means it is formed in the mesoderm, and both sides we can see the mesodermal layer. That means a cavity which is lined with mesodermal layers on the both sides. We can say true body cavity, or a cavity which contains secondary body cavity. That adult, a new cavity which is formed in the adult, we say secondary body cavity. These are the properties of the true body cavity we are talking about. That they should have the secondary body cavity, and they need to line outer and inner lining should be with mesodermal layer. Then only we consider them as true body cavity, and we consider that kind of the coelomates as eucoelomate. Okay, so then let's start reading here. What is given? Let's see here. That coelom or true coelom is a fluid-filled cavity that lies between the body wall and visceral organ. Visceral organ, which is shown here, is the gut. The gut we are shown here. Then all the internal organs in the abdomen we say visceral organ. They should be lined with mesodermal epithelium. That's what we said now. That true body cavity that should be lined with mesodermal epithelium, which we call it as peritoneal layer. Peritoneal layer. Okay. So this peritoneal layer have some knobs. Let's see here. That in case of adult, earlier we have seen somatic mesoderm and visceral or splanchnic mesoderm. As I said to you earlier, that in adult we are going to say it as peritoneum. Okay, so I'll change the color here to show the mesoderm. As I'm drawing the mesoderm with black line. In adult, we have separate terms to see. What are they? Parietal peritoneum. The portion of the peritoneum that underlines the body wall is called parietal peritoneum. So this is parietal peritoneum. So we are talking about this. The peritoneal layer which is present in the ectoderm, inner lining of ectoderm, is called parietal peritoneum or somatic peritoneum. The portion of the peritoneum that covers the visceral organs is called that visceral peritoneum or splanchnic peritoneum. Visceral peritoneum or splanchnic peritoneum. Okay, so this black that I shown to you. So let me say once again, the parietal peritoneum and visceral peritoneum. This is the body wall and this is the alimentary canal. And what we are saying, their inner lining should be with peritoneal layers. That inner lining of body wall can be called as parietal peritoneum. Can be called parietal peritoneum. Outer lining of the gut is called as visceral peritoneum or splanchnic peritoneum. Okay, in coelomates, the visceral organs are suspended in the coelom by the peritoneum. All the visceral organs, kidney, liver, pancreas, spleen, stomach, intestine, everything are going to be suspended over the coelom by peritoneal layer. All they covered with the peritoneal layer. A double layered peritoneum that connects all some visceral organs to the body wall is called mesentery. So, for example, if you take this is the stomach and intestine, and here we have the pancreas, then liver. All these are going to covered with a double layered peritoneum. Double layered peritoneum. That double layered peritoneum is called as mesentery. Mesentery we can see. Then next, let's continue. In some eucoelomates, such as the annellates, the dorsal and ventral mesenteries divide the coelom into paired compartment. We are talking about the earthworm, that these mesenteries are going to form septa, we can see. So compartments, the word they use, that is nothing but septa, which we are going to learn that in detail in earthworm. Okay, so in some eucoelomates such as annelid, the dorsal and ventral mesenteries divide the body cavity coelom into paired compartments. These are the paired compartments which the septa we can see. Certain organs such as kidney, this is very important, just focus here. Kidney of the vertebrates are covered by parietal peritoneum only. That means if you take the kidney is covered by only one layer, that is parietal peritoneum only, that is on ventral side. Only on the ventral side, only parietal peritoneum is present, that is only on ventral side. That we say retroperitoneal organs. The organs lined by it are called retroperitoneum. So before we go these, let us see the remaining also. So I'll show you the lungs. The lungs. Actually, every visceral organ in case of human being, if this is the lung, every human visceral organ will have two layers, like parietal peritoneum and visceral peritoneum. That I said to you in epithelial tissue. Parietal peritoneum and visceral peritoneum. Lungs are covered by pleural parietal peritoneum, plural visceral peritoneum. If you take the heart, in case of heart, if this is the heart is lined by pericardial parietal peritoneum and pericardial visceral peritoneum. Pericardial parietal peritoneum and pericardial visceral peritoneum. Okay, so just little bit confusing area this is. Let me say again, this is the red one, pericardial parietal peritoneum. Then the black one, pericardial visceral peritoneum. And here also, the pleural parietal peritoneum, plural visceral peritoneum. At least in first year, you need to focus that internal organs are completely covered by mesodermal peritoneal layer. Only exception is kidney. Kidney is covered with only peritoneum on ventral side, that is parietal peritoneum. Hence, we call it as retroperitoneal organ. Retroperitoneal organ. Okay, so more animals are given as a space, that compact area as a paragraph. So that's why I'll show you once again that all the issues I'll make you to let that go for once again. So what we said, if you take the transverse section of any organism we are talking about any organism, that we have a mesodermal layer as the inner lining of body wall and mesodermal layer as an outer lining of body wall. Okay, so that is as a common factor they set it over for any kind of organism, including earthworm or any other organism we can take. So we named it. What we named it as parietal peritoneum and visceral peritoneum. We named them as parietal peritoneum and visceral peritoneum. So I am writing here somatic or parietal peritoneum. Okay, so then outer lining of gut, outer lining of alimentary canal. visceral or splanchnic peritoneum. Language actually, peritoneum, when I am using, you need to understand I am talking about the adult individual. When I am using somatic, mesoderm, ectoderm, endoderm, that is embryonic language. Okay, so then next, when take about some more issues are we learn now. Mesentery. What is mesentery? A double-layered peritoneal layer which covers all the visceral organs. Pleura. What is pleura? Pleura is said to be the peritoneal layer which covers the lungs. Pericardium. What is pericardium? That peritoneal layer which lines the heart. We say pericardium. Retroperitoneum. That means the peritoneal layer which covers the kidney. What is the difference between the above and that retroperitoneum is the peritoneal layer which covers the kidney only on ventral side. That is parietal peritoneum. We call it as retroperitoneum. Okay, so let me draw the small diagrams. I'll draw. I'll show you this area. Okay, so this is the heart, and these are the pair of lungs. Okay, so just imaginary diagrams. Okay, lungs and then kidney. Each and every organ is having these kind of peritoneal layers individually in an adult human being, the highly developed animals. As a general issue, we said it over. For that inner parietal peritoneum, inner lining of body wall, outer lining of alimentary canal. But when we come to the human being, we are a little bit advanced in the case. So that's why we have two layers. So let me show you that heart individually have this package. Individually, heart itself have this package. What package? Parietal peritoneum and visceral peritoneum. That parietal peritoneum, pericardial, visceral peritoneum. Then lungs are itself, they have a package of pleural parietal peritoneum and pleural visceral peritoneum. Pleural parietal peritoneum, pleural visceral peritoneum. Then kidney will have only parietal peritoneum as a lining on ventral side that is not surrounded. That you need to understand, that we call the retroperitoneum, we can say. Okay. So a little is there in first year and little we are going to discuss in human physiology. But for continuation, I made it everything here itself, like pericardial parietal peritoneum, pericardial visceral peritoneum of the heart, pleural parietal peritoneum, pleural visceral peritoneum of the lungs, the retroperitoneum of the kidney. So what is mesentery? A double layered which covers all the visceral organs. Double layered peritoneal layer which covers all the visceral organs. We say that is mesentery. Okay, then let me continue. In some eucoelomates. So these are the definitions. As a definition, we need to remember all these things. As a definition, we need to remember. So let me continue. Let's go back. Yes, small technical problem. And now let's continue. Let's continue. We have on the screen. Then retroperitoneal organs. Okay, next, let me continue. During the embryonic development, the eucoelomates, the blastocoel is replaced by true coelom derived from the mesoderm. I think, so when I say like this, you people will be much visualization you will get actually. But that because I already explained everything to you. The coelom, just we are reading. During the development, I have some technical. So what we said that during the embryonic development of the eucoelom, the blastocoel is replaced by true coelom. True coelom means what we said that blastocoel is considered as primary body cavity. Now whatever the true coelom we are going to say, that is said to be as secondary body cavity that derived from the mesoderm. So the true coelom is also called secondary body cavity. The true coelom is also called secondary body cavity. In the eucoelomates, mesoderm lines the both body wall and the walls of the visceral organs. So visceral organs become muscular. That we said earlier. That mesoderm is wherever the mesoderm is present, their muscles can be seen over. That's the reason in pseudocoelomates, elementary canal is non-muscular. Okay, then let me continue. About as the wall of the gut becomes thick and muscular, digested food and other nutrients cannot easily diffuse from the lumen of the gut into the coelom or fluid, which is actually possible in case of pseudocoelomates. In case of nematoda, we said that elementary canal is non-muscular. That's why that nutrients are getting diffused directly because of non-muscular elementary canal is generally thin, isn't it? So that's why easily they diffused over. But in case of true coelomates, mesoderm is also towards the gut. That's why elementary canal is going to be muscular. Like if you take earthworm, buccal cavity, pharynx, esophagus, everything is muscular. Okay, in case of human being also, just I am taking one example. In eucoelomates, elementary canal, because of the mesoderm, it become muscular and thick. So that's why the nutrients cannot easily diffuse from the lumen of the gut into the coelom, like pseudocoelomates. Then circulatory, do remember, this is a continuation of the previous class. These kind of things are discussed earlier in the previous class. So coelom is actually, explanation is over. Now I am showing how it is given in the NCR, the textbook. Okay, so then the circulatory system, that means as like pseudocoelomates, it is not possible diffusion of the nutrients outside directly from the gut of elementary canal. That's why there a new the circulatory system is developed in the eucoelomates to overcome that problem. Based on the mode of formation of coelom, we classified the eucoelom into two types. Okay, that is about shageocoelomates and enterocoelomates. Shageocoelomates, enterocoelomates. So let me come for the shageocoelomates. I do remember you once again about the shageocoelomate story. We said by the splitting of mesoderm, mesoderm develops, mesoderm fuses, and mesoderm reaches one side to the ectoderm side and other side towards the endoderm side. We call it as the primary body cavity is closed, and that leads to the secondary body cavity. This is the blue one I am showing. That leads to the secondary body cavity. Okay, so now let's start. Let's start reading. Animals in which the body cavity is formed by the splitting of mesoderm are called shageocoelomates. Example, Annelida, Arthropoda, Mollusca are the shageocoelomates in the animal kingdom. All the shageocoelomates are protostomian, isn't it? The protostomia ends, and they show the holoblastic, spiral, and determinate type of cleavage. Because of our kingdom Animalia series, these things are not at all new for you. Okay, so what's new? That 4D blastomere or mesentoblast cell of the early embryo divides to form the mesodermal blacks between the ectoderm and endoderm that replace the body wall. So I'll remind you once again here, this is the gastrula stage. In the gastrula stage, we said the outer layer is ectoderm and inner layer is endoderm. Inner layer we call endoderm. This is the endoderm. And from here, we said the address also. What is the address? At the rim or margin of blastocoel, where ectoderm and endoderm are continuous each other, mesodermal cells are getting originated. Okay, which mesodermal cell they are saying? The 4D blastomere or mesentoblast cell of the early embryo divides to form the mesodermal blacks between the ectoderm and the endoderm and replaces the blastocoel or primary body cavity. That appears, the split that appears in each mesodermal block leads to the formation of shageocoelom, split coelom. Shage means splitting. In the annulates, the functional body cavity That means perivisceral cavity. Functional body cavity of annelid is perivisceral cavity is Schaeger-coelom. It is in the form of series of paired coelomic cavities. But we have an exception. Basically, we said Annelida, Arthropoda, mollusca are Schaeger-coelomate. But in case of Arthropoda and mollusca, the functional body cavity that lies around the visceral organs filled with blood and it is called hemocoel. That is an exceptional issue here. So we just talked about true coelom. That's over. But if you take the cockroach as an example here, cockroach, in case of cockroach, open vascular system is present. The body cavity of cockroach is called hemocoel. Body cavity of cockroach is said to be hemocoel, and blood filled in the cockroach is called hemolymph, white color blood. Okay, I am talking as a representative of phylum Arthropoda. It is formed by the fission of embryonic blastocoel with some true coelomic spaces, and the tissues are directly bathed in the blood. Okay, that should be elaborated actually. This is a little confusing area. So that should be elaborated. So let me elaborate this. I am going for whiteboard. So I am going to elaborate this area. Then that should be little bit elaborated in case of phylum Arthropoda and mollusca. By choice, that they should have the Schaeger-coelom, isn't it? Schaeger-coelomates they are. Okay. But that Schaeger-coelom, what is Schaeger-coelom? True coelom. So whom we call true coelom? Those who have true body cavity. Who is called true body cavity? Those who have secondary body cavity, they are called true body cavity. How do you recognize that is secondary body cavity? That should be lined by mesodermal layers, lining of mesoderm, or in adult case, lining of peritoneum. So what we are saying here, multiple issues in single statement they explain. That's why I'm talking. First of all, the blood of cockroach is called hemolymph. So why we need to see here, why it is called hemolymph, that you need to understand. Why it is called hemolymph? Blood of cockroach is white color. That's why we said it as hemolymph. Then where it is? It is in the hemocoel, body cavity. Hemocoel. Body cavity of cockroach is called hemocoel. Why it is called hemocoel? Blood-filled body cavity. Body cavity filled with the blood. That's why it is called hemocoel. Okay, so what's the news? How it related to coelom? That's what we are talking. This is not lined by mesodermal layers. That means this is embryonic blastocoel. We know that those who have embryonic blastocoel, we say that as false body cavity. So why don't you join them in pseudocoelomata? No. Actually, true body cavity is located in reproductive and excretory organ. That means they have the personal exception here. True body cavity. Whom we call true body cavity? Secondary body cavity. True body cavity or secondary body cavity are body cavity lined with mesodermal layers. Body cavity lined with mesodermal derivatives is at only reproductive and excretory organs. At reproductive and excretory organs. Actually, we have little explanation in animal kingdom next chapter. So by taking chance, everything I am going to say here. True body cavity or secondary body cavity or body cavity lined with the mesodermal derivatives, nothing but peritoneum, is at only restricted at only reproductive and excretory organs. Only reproductive and excretory organs. What about the remaining body cavity? The remaining body cavity is false body cavity. Whom we call false body cavity? That primary body cavity. Primary body cavity is called false body cavity. So focus here. The body cavity of cockroach is hemocoel. What is hemocoel? That's what I am saying. What is hemocoel? Hemocoel is false coelom. False coelom. False body cavity. Hemocoel is false body cavity. Okay. So that's it. Hemocoel. That all the different issues in one place. They said that's why the problem here. Okay. So why it is called hemolymph? Why it is called hemolymph? Blood of cockroach is white in color. Blood of arthropoda, cockroach is white in color. We call hemocoel. So why it is called hemocoel? Body cavity, blood-filled body cavity is present there. That's why we say that as hemocoel. So what is hemocoel? That will come to our topic. What is hemocoel? Embryonic blastocoel or primary body cavity. Embryonic blastocoel or primary body cavity. Okay, so that's why we say this is the confusing line. So that's why I'll go with the next page. As some definitions wise, I'll show that definitions wise. Number one, hemolymph. Who is called hemolymph? White color blade of cockroach. White color blade of cockroach is called hemolymph. Okay, white color blade of cockroach is called hemolymph. Okay, next. Who is called hemocoel? Whom we call hemocoel? Hemocoel.
Body
Body
cavity.
cavity filled with blood is called body cavity filled with blood. In future also it will be useful for you. Body cavity filled with blood is called hemocoel. Okay, so that is called hemocoel. So, what body cavity? Hemocoel
Four.
is primary body cavity or secondary body cavity? Hemocoel
Primary.
is a
Primary.
primary body cavity. Hemocoel is a primary body cavity. So that's why, what do you say?
False
Primary
body
body
cavity.
cavity, we say yes, false body cavity, isn't it? So then how can you consider arthropoda is as a having Shageosilome? Yeah, secondary body cavity or true body cavity, secondary body cavity or true body cavity or body cavity lined with peritoneal layer, or that means or body cavity lined with the peritoneal layers is restricted with only at reproductive and excretory organs. Secondary body cavity or true body cavity or body cavity lined with peritoneal layers is restricted at reproductive and excretory organ. At least there is a cavity, peritoneal cavity, but most of the body cavity is hemocoel, and that cavity is primary body cavity, embryonic blastocoel. That was an exception, personal cases. That if you want to see the exact Shageosilome, our earthworm, Annelida is there a best example, but in Arthropoda and Mollusca, they have some exceptions like this. Okay, so let me continue the text here. Let me continue. That is all about the story of Shageosilome. And then now moving on towards the enterocoelom. Here you see the pouches are formed. Enterocoelom, pouches of Archenteron. Animals in which the body cavity is formed from the mesodermal pouches of Archenteron are called as enterocoelomates. Echinoderm, hemichordates, and chordates are the enterocoelomates. Echinoderm, hemichordates, and chordates are the enterocoelomates. In these animals, mesodermal pouches that evaginate, you see here, this is the area of evagination, that evaginate from the wall of Archenteron into the blastocoel are fused one another to form the enterocoelom. I just shown with some more clear diagrams here in the as in the form of transverse section, like the pouches are formed from the Archenteron, and then the pouches are developed. Pouches are developed. So let me take another color. So here it is. The pouches are developed. Then pouches are fused. Pouches are fused, and one reaches to the ectoderm side and one reaches to the endoderm side. That was the total story we learned. So finally, what is the final conclusion? The mesoderm reaches towards the ectoderm side and towards the endoderm side. the true body can. Okay. Then all the enterocoelomates are deuterostomes. They show radial and indeterminate type of cleavage. That's it. This is the total story of enterocoelomata. Okay. Before I continue to the advantages of coelom over pseudocoelom, before I continue, so there I'll go with what is 4D blastomere. Not much necessary here, but that name came, that's why let us learn little bit interesting item even. Determinate type of cleavage. Where do you see the determinate type of cleavage? In which phyla? Annelida, Arthropoda, Mollusca are having determinate, but if you say total determinate type of cleavage, indeterminate type of cleavage, did you remember? Belongs to which division it is? No sound.
Protostomia.
Ah, very good. Division Protostomia. That means from phylum Platyhelminthes to that mollusca, isn't it? Phylum Platyhelminthes to mollusca. That means our Annelida, Arthropoda, mollusca is also as a part of determinate type of cleavage. So why we are looking for the determinate type of cleavage? There we said like this. In case of Schaejocoelomates, we named the cell as a mesoderm here like this. This is the ectoderm and this is the endoderm. And as I said to you, the 4D blastomere, 4D blastomere or mesentoblast cells are going to form the mesoderm and the split. That they said like this. Okay. So what is 4D blastomere? So to know what is 4D blastomere, you need to know what is determinate type of cleavage. If you know what is determinate, you will find what is 4D blastomere. We learned actually what is determinate type of cleavage. The fate of blastomeres will be decided early in the embryonic development. I will make you to remember once again, fate of blastomeres during embryonic development, fate of blastomere will be decided very early in the embryonic development, decided early in the embryonic development. That we said it as determinate. So how do it related with that? Now let us see here on next screen. I'll show here how does the determinate is correlated with the Schaejocoelomate. During embryonic development, when zygote, one cell, two cell, four cell, here, when it comes to the four cell stage, the fate will be decided. That's what we said it as, determinate. Every cell will decide, I'll become something likewise. That's why we call it as determinate type of cleavage. The fate of blastomere decided early in the embryonic development. Our scientists, our embryologists, they started giving naming for this particular. That means at 4 cell stage, each cell is named as A, B, C, D. If the 4 cell stage divides into 8 cell stage, they'll give further names again, like 1 capital A, 1 small a, 1 capital B, 1 small c, 1 capital C, 1 small c, 1 capital D, 1 small d. That means every cell is again named like this at 8 cell stage. Okay, so why they are giving names? They are following the cells, what they do exactly. Okay, this is the 8 cell stage. Then what about 16 cell stage? Let me show you. 2 capital D, 2 small d. 32 cell stage, 3 capital D, 3 small d. 64 cell stage, 4 capital D, 4 small d. Came to our destination. 4 small d is responsible for the formation of mesoderm in Schaejocoelomates. In Schaejocoelomates. 4D blastomere. This is the story of 4D blastomere. Actually, these things are not necessary for us. That's why I just came like this. 4 cells. And this is 8 cell stage. This is 16 cell stage. 32 cell stage. 64. You have to remember all 64 names will be given there. In that, our 4D blastomere is responsible for this. Okay, so to avoid the confusion, I'll take only one here, only one as a full, not much required, but interesting thing here. This is the 4-cell stage. So I'm moving only with one capital D, one small d. That divides. That divides two capital D, two small d, three capital D, three small d, four capital D, four small d. So that every dividing cell will be given a name here like this. Here these are also having names: one D1, one D2, one D11, one D12. That one D21, one D22. Okay, this pattern will go on, but we require 4D blastomere. That's why I came in this series. Okay, so that means 4-cell stage is having names A, B, C, D. 8-cell stage is having names 1A, one small a, one capital B, one small b, one capital C, one small c, one capital D, one small d. 16-cell stage, every cell has their own name. 32 cell, 64 cell, every cell has their own name. So in that aspect, that 4D blastomere in case of Aceolomates, due to the determinate, our embryologists are given starting name. Imagine this is a 64 cell stage, and there is a cell called 4D blastomere, and that 4D blastomere is going to form the total mesoderm. That's what we said, determinate. 4D is dedicated for that mesoderm. That's why we call it as determinate type of cleavage. Okay. So just for general knowledge, now let's say here, just we have only one line in the text here, one line, that is, here it is. In Aceolomates, they said it about 4D blastomere. That why 4D? What is the name of 4D? That simply I can say 64 cell stage, the name of one cell is 4D blastomere. Okay, so that we can see. Okay, then that just for information we provided. And now let's give the conclusion to the Coelom chapter. Let's give the conclusion to the Coelom chapter. Advantages of Coelom that we need to go through. Advantages of Coelom. Let me erase this. So, keyword to be remembered for Aceolomates, splitting of mesoderm. For enterocoelomates, pouches of archenteron. So finally, let's close this area. Advantages of coelom over pseudocoelom. Okay, visceral organs of eucoelomates are muscular, that we discussed now. That elementary canal of true coelomates is muscular. Visceral organs of eucoelomates are muscular because they are associated with mesoderm and can contract and relax freely independent of the muscular movements of the body, while in the coelomic space, what do you say? Peristaltic movements of the elementary canal. So that means for that we have to take a good example to understand a pseudocoelomate and a pseudocoelomate. So let me take a pseudocoelomate first of all. We have to take the round worm. Let me take the round worm this side, and this side I'll take earthworm. Earthworm is a representative of phylum Annelida, isn't it? Then, in case of earthworm, elementary canal, buccal cavity, pharynx, esophagus, gizzard, more muscular, gizzard, then stomach, intestine. Have you seen the difference here? The elementary canal is much thicker because of muscles. Here, the elementary canal is very thin. In this case, elementary canal is thin because of non-muscular nature. If there is no muscle, that's why this elementary canal movement, that individually, independently, it cannot move. This elementary canal independently, it won't move. Why? Because there is no muscles. Okay. Then here, there is a possibility of independent movement of the elementary canal. When the food is dropped here, this area will come into contract, food will be forced here. This area will be contract, food will be forced here. What do you call this kind of movement? Peristaltic movements. Rhythmic movements of the elementary canal are said to be peristaltic movements. So that is possible in case of true coelomate. And when the elementary canal is moving, the movements of elementary canal independent of the movements of the body wall. That you can feel itself, that you can feel. That means when you are taking food material, that from the esophagus to the stomach, stomach to the intestine, food will move, isn't it? It will move by your body movements, or they have independent movement. They have their own independent movement from esophagus to the stomach. You are making snake dance to transport the food. No, your elementary canal performing the snake dance. That means what I am talking, peristaltic movements. Okay, so these peristaltic movements are possible only in the true coelomates because elementary canal is muscular in them. So that we have two statements with this. The movements of elementary canal are the dependence of the movements of body wall in pseudocoelomates. The movements of elementary canal are independent of the movements of the body wall in true coelomates. Whatever the true coelom, annelida to chordata. Okay, next. The gametes are released into the coelom in some invertebrates which do not have gonoducts and in female vertebrates. That means that is also a kind of possibility here. Body cavity is present. That's why if you take annelida, that this is the ovary of annelida, they release the eggs into the body cavity. That means body cavity is present. That's why they can release. In case of human, the human being also, from the ovary, the ovum or secondary oocyte, Graafian follicle, will be released into the body cavity. Okay, that possibility is there. We are talking about. Okay, female vertebrates only. Male vertebrates, sperms are transported by epididymis. They have a special duct, a series of sequence of ducts. In non-chordate, coelomic fluid receives excretory products and stores them temporarily before their elimination. That means having body cavity, that is in case of non-chordates, that means in invertebrates like earthworm, cockroach, and frog. Earthworm, cockroach. The frog is a vertebrate. In non again a little disturbance. So need to go there. So in case of non-chordates, there is a possibility of excretory products. Coelom presence helpful for the storage of excretory products before their elimination. Okay. Next, in eucoelomates, the mesoderm comes into contact with the endoderm of the elementary canal. In contact with the endoderm of elementary canal, it causes regional specialization of the gut, such as development of gizzard and stomach. That what I shown to you, that the regional specialization of the gut, which is referred to as primary induction. This kind of regional specialization is called primary induction. In case of pseudocoelomate, due to the absence of such a contact between the gut and mesoderm, the wall of the gut does not show complex and highly specialized organ. That means that if you take a pseudocoelom, and I'll show you the pseudocoelom during embryonic development, what we say that mesoderm went only towards the ectoderm side, isn't it? Mesoderm went only towards ectoderm side. But in case of true coelomates, mesoderm is having that went to the ectoderm side, as well as also it is interacting with the endoderm. That's why elementary canal became muscular. That's why this elementary canal became muscular. The association of the mesoderm with particularly endoderm. Association of the mesoderm with endoderm. So have you seen the difference? This is a pseudocoelomate and this is a true coelomate. In pseudocoelomate, mesoderm is not coming in contact with the endoderm. But in case of true coelomate, mesoderm is having connection with the endoderm. So that connection we call, I'll make you out back again. This is the mesoderm, and which is having connection with the endoderm during embryonic development. We call this as primary induction. What is the advantage? This elementary canal become non-muscular. This elementary canal become muscular. That is the advantage. That we are seeing from the beginning. Elementary canal is non-muscular, elementary canal is muscular. Okay, so that's it. Okay, let's see this in non-chordate coelomic fluid receives excretory products and stores them temporarily before their elimination. Actually, in earthworm, we can see that excretory products are released in the body cavity. So then after, they'll be collected by excretory organs. That is possible. In case of cockroach, completely through the body cavity, they go to the excretory organ. So simply that we'll discuss when we go to the earthworm and cockroach, we'll go and discuss them in detail. So just we have a line here. So that's it. Finally, we are closing the coelom here. We are closing the coelom. The presence of a simple thin-walled gut in the nematodes allows easy diffusion of the digested food into the pseudocoelomic fluid. That's what we are saying from the beginning. From the beginning, we are going to say like this. Elementary canal of nematoda is non-muscular. So non-muscular elementary canal disadvantage is it cannot show peristaltic movement. Advantage is absorption of the nutrient directly possible into the coelomic fluid. Okay, the compensating the absence of circulatory system. That we said in case of true coelomate, elementary canal is muscular. So earthworm I am taking example. I'll show you once again that elementary canal is muscular. Due to the muscular nature of the elementary canal, that mouth, buccal cavity, pharynx, gizzard, likewise thicker elementary canal, our said thing, that means nutrients can't diffuse directly. That's why there is a need of circulatory system, development of circulatory system due to this only that they want to see. Okay, so that's it. Yeah, after this is a just formality for us. We are masters in this area. The kingdom, levels of organization, symmetry, and body cavity, phylum, wise. Cellular level of organization is there in kingdom Animalia, subkingdom Parazoa of phylum Porifera. And tissue and organ system grade of organization is seen in kingdom Animalia, subkingdom Eumetazoa. Eumetazoa further classified into grade Radiata with radial symmetry, grade Bilataria with bilateral symmetry. Radial symmetry is there in Nidaria and Ctenophora. Bilataria. The division by the grade Bilataria is classified into two divisions, protostomia, deuterostomia. In case of protostomia, acoelom, pseudocoelom, and schizocoelomates are present. Acoelom, without body cavity, acoelom, phylum Platyhelminthes. With false body cavity, pseudocoelom, phylum Nematoda. Then the Schizocoelomate, true body cavity from the splitting of mesoderm, Annelida, Arthropoda, mollusca. Then true body cavity, the division deuterostomia is having one subdivision, that is enterocoelomata with true body cavity from the pouches of Archenteron with three five, Echinodermata, Hemichordata, and chordate. So this is all about the coelom, all about the coelom.